Okay, hopefully that has worked and you can all see my slides now. So as I mentioned, I'm just going to cover a little bit of the portfolio of research that we have at Anthony Nolan, specifically focusing on our patient donor project, which is one of Anthony Nolan's longest running research studies and the one that really impacts on how Anthony Nolan functions, some of the decisions we make around donor selection, and how we pick donors for our patients and what kind of donors we want onto our register. So I'm going to start off by being a little bit rogue and start off with my acknowledgements, because I think that it's really important to acknowledge that we couldn't do the research that we do without the support not only of the patients and donors who consent to be part of this study. They're absolutely vital in the success of this project, and to improving transplant activity and donor selection for patients and donors who come in the future. But really, I'd like to say a very big thank you to all of you, and to everybody who raises money for Anthony Nolan, who raises awareness about Anthony Nolan, and who recruits individuals to our register. We really couldn't do the work that we do without you, and particularly our research. It's so vital that we do some of the work that we're doing, and that we are actually continuously improving things for patients, and we couldn't do that without your support. So a huge thank you up front to all of you for the fantastic work that you do as part of Marrow. I'd also just like to quickly thank all of the UK transplant centres with whom we collaborate, and my colleagues in various organisations, both Anthony Nolan, BSBMT, CT, and the CIBMTR as well. So the patient donor project that I've mentioned, it's really been set up because we know that there are multiple different factors, clinical factors, that can affect the outcome of stem cell transplantation. So we know that there's a balance between the clinical factors to do with the patient and the donor, the genetics of the transplant. So for example, the tissue typing genes, how we match patients and donors. And when we talk about tissue typing, we're actually referring to a family of genes known as HLA. And I'll explain a little bit more about HLA further down the line. But we're also very interested in how factors such as non-HLA genetics impact on the transplant outcome. And we know that in combination, by mixing all of these things together in the right way, of having the right balance of an algorithm, we should hopefully get successful transplants for everybody. So the Anthony Nolan Patient Donor Project, as I mentioned, it's one of Anthony Nolan's longest running research studies, and it aims to determine how genetic and clinical factors to do with the transplant itself, and specifically to do with donor matching, impact on patient outcome. And we actually collaborate with every transplant centre in the UK that performs allogeneic unrelated donor transplantation. So there's about 37 centres across the UK that we collaborate with every day. This is the team. I don't do this on my own. It's very much a team effort, and this is only the research team. There's a lot of people around Anthony Nolan who support this project. But in terms of those of us in the Research Institute, the project's led by Professor Stephen Marsh, who's our Director of Bioinformatics, and also has the support of Dr. Chloe Anthias, who's our Medical Director. And then I have a fantastic team of individuals working in the lab. So we've got Tom, Johnny, Charlotte, and Jack at the moment, who are working with us to actually do the research on the samples and process the samples as they come into the Institute. And they really are an amazing bunch of young scientists who are going to go to amazing places. I think they're really fantastic. And again, the work that we're doing and the data that we're generating really comes from them. So our project aim is to recruit every patient in the UK that receives an unrelated donor stem cell transplant from a UK donor, and to recruit the corresponding UK donor as well. And the only reason that we limit this to UK patients and UK donors is logistical. It's very difficult to have the ethics and the permissions to do studies to recruit individuals from lots of different countries. So that's the only reason we limit this to UK. We collect a blood sample from the patient prior to their transplant, and from the donor as well at the point that they have their medical prior to donating. And then we need to have clinical outcome data as well, and we collect that data through a collaboration with the British Society for Blood and Marrow Transplantation and Cellular Therapy, so BSBMTCT. When we've collected the blood samples, we receive them back in the lab and then we process them. So for anybody who hasn't extracted DNA before, this is what DNA looks like when it's been freshly extracted, or partway through its extraction process. So this little sort of cloudy white clump here in the bottom of the tube is actually DNA that's partway through its extraction process. What we also do with part of the blood sample is to layer it on top of a very concentrated sugar solution, which is this clear liquid at the bottom here in the tube. And then we spin that using a centrifuge. And what we're able to do is actually separate out here, if you can just see this sort of cloudy layer here, this is actually our white blood cells. We're able to separate those out and store those for downstream experiments. Sadly, in 2021, it would be a little bit remiss of me to not mention COVID, as much as I wish I didn't have to, but COVID-19 has really had a big impact on our research work. So for the first time in 24 years, we actually had to pause the patient donor project. We stopped recruiting anybody to that study during the period really of the first lockdown between March and July of last year. On top of that, our research institute was closed for the first time in our history. However, we did aim to have everybody back in the lab by the 1st of July, and we were successful in that. And we've had projects back up and running since then, and the teams have been coming in ever since in a reduced capacity. However, the impacts on our research really been considerable. It can take many months for a research project to pick back up. And in terms of the patient donor project and the number of individuals that we're recruiting for the studies, it hasn't really reached the pre-pandemic levels at this point. So this is just giving you an idea here. Each of these lines here represents the number of requests that we've had in a year. And by requests, we mean the number of eligible transplants that we've been able to try and recruit patients and donors from. You can see that over time, we've had a steady increase, particularly around 2012, when we first started with the Aligned UK registry. So that's partnering with other registers in the UK to form an Aligned UK register. But obviously 2020 had quite the impact on the number of individuals we were able to recruit. And although in 2021, it seems we're going to do better than we did last year, you can see that the numbers haven't quite recovered to the levels they were pre the pandemic. But we're hopeful that this is a temporary measure and we should start to see recruitment increase in the next few months, or hopefully going into the next year. One of the limiting factors for us when we are looking to recruit individuals to a study is that we need to have both the patient and the donor blood sample in order for us to be able to take those pairs and move them through to further analyses. And what you can see here from this graph is that the blue line represents the number of donor samples that we've received, and the black line represents the number of patient samples that we've received. And you can see that for every single year, the number of donor samples is far greater than the number of patient samples. So actually recruiting patients to the study is much more challenging than recruiting donors. And there's lots of reasons for this, including the fact that a lot of these patients are being asked to donate blood for treatment-related purposes. They don't want to be prodded and poked any more than they already are. And also particularly for pediatric patients, parents often don't want their children to have blood taken unnecessarily. So there's quite a few challenges around this. But on the whole, we do get a really positive response from patients, and they do want to be part of our research as much as they possibly can. So I mentioned the three different factors that we believe come together in terms of having a the best possible transplant outcome. And we believe that the HLA factors, the tissue typing genes, are some of the most important in terms of predicting good transplant outcome. So this is just some cartoon diagrams of the HLA genes themselves. As I said, the genes themselves encode the molecules that make up our tissue type, and their function is to help us recognize what is us from what is not us. So if you are, like me, suffering from a little bit of one of the colds that are going around at the moment, what's happening is that your HLA genes are presenting parts of the virus that you've got to your immune system, and hopefully your immune system is kicking in and helping your immune system to knock out that virus and to help you to recover, to mount that immune response. So where the tissue typing and the HLA molecules are really important in terms of transplantation is that we want to be putting in molecules into the, we want to be putting in cells into the patient from the donor, where the HLA molecules are not going to see patient tissue as something that isn't self. We want to match individuals as closely as we possibly can so that their tissue isn't recognized as non-self, because that leads to complications such as graft versus host disease. And we really want to avoid that as much as possible after the transplant. So there are two main classes of HLA genes. They're known as class 1 and class 2. The class 1 molecule, which is just presented here, is encoded by a single gene, which encodes this yellow structure just here. And actually it requires a secondary protein known as beta-2 microglobulin, which is encoded on a completely different chromosome from HLA to stabilize that molecule. The class 2 molecules are encoded by two separate genes. So we have the alpha gene, which is encoded in pink here, and the beta gene, which is in blue just here. But although they're structurally slightly different, the way that they function is very similar. They tend to form this groove, which is known as the peptide binding groove just here. And that's where parts of the viral protein or the bacterial protein, or in the case of transplantation, maybe parts of patient tissue are expressed to the immune system so that they know whether they can recognize these as self or as non-self and mount immune responses to it. And so in terms of transplantation and matching and the work that we do to type individuals, a lot of attention has been focused on these parts of the gene because of their functional importance in terms of transplantation. If we look how this works in a family itself, the HLA genes are codominantly inherited. So both copies that we inherit from our parents, one from our mum, one from our dad, they're both important, and we need to consider both copies that we have. So here we have Homer, and this is a very simplified version. I'm just considering 3 of the HLA class 1 genes, but you can see that he's inherited one copy of his tissue type on one chromosome and a second copy on his second chromosome. Here we have Marge. Marge also has 2 different copies of her HLA genes, but she's inherited different tissue types on each of her 2 different chromosomes. And when they have Bart, you can see that he has inherited this copy from his father and this copy from his mother. Lisa comes along and she's inherited the same copy of her HLA genes from Homer, but a different copy from Marge, and so on and so on. And I do know that these 2 are not Simpsons, but it's very difficult to find a famous family with 5 biological children, so you'll have to forgive me for this one. In this case, here we have Bart, and Bart needs to have a transplant. So the first thing that we do is actually search through the family to see whether we can find an individual who has inherited both copies of the same haplotypes from each chromosome from their mother and father. And in this case, we have Martin, who happens to have inherited the same 2 copies. So he would be the ideal match for Bart in this instance. However, for many patients, they don't have the choice of a family donor, a related donor, and that's where Anthony Nolan comes in. And we have a wonderful group of volunteer donors who are very generous and would volunteer to donate their stem cells to a patient in need. Apologise, I can hear a dog barking, but I'm not sure where it's coming from. We also have an umbilical cord blood bank, and we have here umbilical cord blood cells, which are an incredibly vital source of stem cells for patients post-transplant. And they are really, really important, particularly in cases where we can't find an HLA-matched adult unrelated donor for individuals. So that's also an incredibly important resource for our patients. So I've told you that HLA matching is important in terms of transplant outcome, but here's the proof. So this is a graph that looks at matching between 5 different genes. So we call that a 10 out of 10 match. That means that they're matched for both copies of the 5 genes that we are considering here. When we refer to a 9 out of 10 match, we mean that there is a single mismatch in one of the HLA genes between the different tissue types that they are there, and so on and so on. 10 out of 10 matching has been the gold standard for matching in the UK and in Europe for many years. And some of the work that we've done that I'll talk to you a little bit about later on has actually challenged this to see whether this is really the gold standard. But it has been for many years, and it is generally what we're looking for when we are trying to find a donor for an individual. So what you can see here is the blue line, which represents the 10 out of 10 matched donors. That line has significantly higher overall survival than those individuals who receive a 9 out of 10 match, or anybody who receives either an 8 out of 10 match or less than. So really we want to be aiming for as great a match as we possibly can. But this is challenging in itself because the HLA genes are hyperpolymorphic. And what I mean by that is that there are many, many different thousands of different tissue types that we have to be able to identify in both our patients and donors, but also match for them to get that 10 out of 10 match, so that they're matched for as many of the HLA genes as we can. And this proves quite challenging, particularly in the laboratory. So many years ago, when we would do HLA typing, we would use a method known as serology. And what we did here was actually to use antibodies that were specific for different HLA types. And we would use them to stain the outside of white blood cells to look to see whether they had the specific HLA type that the antibody was specific for. And they would fluoresce either green or red, depending on whether they actually express that particular HLA type or not. And this was a really useful technology. In fact, it's still used today. It's one of the few ways that we have that we can actually see whether HLA molecules are expressed on the surface of cells or not. However, we've now moved to using DNA typing methods instead of this serological approach. And the hyperpolymorphic nature of HLA is primarily responsible for this shift in the way that we're typing. So as an example of that, using serology, we were able to type somebody as being HLA-A2, and that was all we knew. However, what we know now is that there are over 6,400 different subtypes of A2 alone, and this is only about a fifth of those different subtypes. I tried to squeeze them all onto one slide, and I was down to font size 2, and it was getting a little bit ridiculous. But this is about a fifth of them. So, and this number is increasing almost daily. And so we need to be able to use DNA technology to differentiate between all of these different tissue types. But even some of our DNA technology is finding that challenging, and we're having to move to new types of technology to be able to characterize each of these individual tissue types. In our labs, we've recently implemented our 3rd generation sequencing platform known as the Pathfinder Sql System. This is a picture of my team just down here where they were doing their virtual training earlier this year and did an amazing job despite being in the middle of the second or third wave of COVID back in February of this year. What this new technology is allowing us to do is to sequence through the whole of the HLA genes in isolation, which is hugely beneficial. First of all, we're capturing all of the information about the different HLA genes, but we're also phasing the results. So we are able to determine from all of the polymorphism that we see, the cis-trans combinations on the two different strands of the DNA, the two different haplotypes that we have. We're able to phase all of the polymorphisms across them. So we get definitive HLA typing really for the first time. So now that we have all of this technology and we're able to type HLA to a resolution that we've never been able to do before, does it actually result in any benefits for the patients? So this is a question that we've been asking, and the technology is now allowing us to answer. And as I mentioned before, the 10 out of 10 match for patients and donors has been the gold standard for many years. And we wanted to challenge that to see whether typing at this new resolution for HLA actually led to a difference in what we should be aiming for when we're finding donors. So we used our patient donor project cohort to type a cohort of 891 donor and recipient pairs using our third generation sequencing technique. These individuals were transplanted at UK transplant centres between 1996 and 2011, and included a cohort of both adult and paediatric patients, all of whom had a blood cancer, so they all had a hematological malignancy. The median patient age was just over 41, although the range was significantly more than that, and quite a considerable range as well. And the disease breakdown was probably quite typical of the era that the transplants covered. So predominantly AML and ALL. We did have quite a few CML patients, that's chronic myeloid leukemia. We don't tend to transplant people for that disease anymore, or very, very few cases. It's actually a fantastic example of where drug development has actually meant that we don't need to transplant patients as much as we used to. And also, again, reflective of the era that this study took place, we're about half of the cohort having a transplant from peripheral blood stem cells, so collection of the stem cells rather than collection of bone marrow, which has tended to increase in more recent years. So forgive me, this is a very busy slide, but I will talk you through it. What we have here is the results of our third generation sequencing on and the impact on the matching status for the individuals. So in this column just here, this tells us how well matched we believe the individuals were at the time of their transplant. And then across the top here is the new level of matching that we found them to be once we were sequencing more of the gene. So one of the first things that we noticed is that for those individuals where we knew that they had mismatches, the number of unknown mismatches increased. So for those individuals here that were a 12 out of 12 match prior to their transplant, so that includes the 6th HLA gene that we believe to be important, just over 75% of those remained a 12 out of 12 match, with the remainder becoming less well matched. But as we increased the number of known mismatches, the number of individuals that remained that well matched decreased, which is not entirely surprising. It was what we had thought was always happening. So the number of known mismatches was actually indicated that there were more unknown mismatches. So one of the first things that we wanted to do was to actually have a look at these individuals that were matched at a 12 out of 12 level. And we wanted to compare those that where we thought they were 12 out of 12 and see how survival differed for those that remained at 12 out of 12 and those who actually had additional mismatches that we didn't know about at the time of the transplant. And that's what we can see here on this graph. So the line in blue at the top here are those individuals where after recharacterization using our 3rd generation sequencing technique, we still found that they were a 12 out of 12 match. And the green line here shows those individuals where we thought they were 12 out of 12, but actually found that they had mismatches. And you can see that there was a significant difference in overall survival for these 2 different groups, and with quite a considerable difference in overall survival at 5 years as well. I mentioned to you previously that we wanted to challenge the idea of a 10 out of 10 match being the best possible match. And this graph here just includes those individuals who are not just a 10 out of 10 match, but a 10 out of 10 match at our 3rd generation sequencing level. And what you can see here is that we have been able to stratify the outcomes, the probabilities for all of these individuals by anywhere up to 45%, using not only the inclusion of this 6th HLA gene, but also the inclusion of an additional characteristic known as Cmv matching for patients and donors. And I'll come back to Cmv matching a little bit later on. But what you can see is really that if we start to use new technology and new ways of characterizing HLA genes at this higher resolution, we are actually able to really challenge what has been thought previously and redefine what the gold standard for matching for patients should be in more recent eras. One of the questions that we've always wanted to answer for our transplant community is, are all mismatches equal? So we know that for some patients, they don't have the choice of having the perfect match. So if they have to have a mismatched transplant, where should we be mismatching and have the least impact? And what we've been able to show is that not all mismatches are equal. So this is an example here of the survival curve that you've seen previously on previous slides. And you can see here in the blue line, these individuals have the 10 out of 10 match in this case, which we believe to be at this point the most important. But what you can actually see here is that there is a difference between the red and the green line between those individuals who are all a 9 out of 10 match, but the difference is where they're matched. So the red line here are 9 out of 10 match, but they are matched for a gene known as HLA-B. And in the green line, these individuals are also a 9 out of 10 match, but they happen to be B mismatched. And so what we've been able to show here is that if we have to pick a 9 out of 10 matched donor, we really want to try and be finding them an HLA-B match wherever possible to help get some of the survival benefit of that particular mismatch. This is data that has a look at this 6th HLA gene that I've mentioned, HLA-DPB1. And what we've been able to do with DP, and Anthony Nolan has worked on this in collaboration with groups in Germany, is the development of a model that talks about permissive mismatching for the DP gene. So here, what we've been able to show is, although we believe matching for DP, so in this case, that means a 12 out of 12 match is the best possible outcome, actually permissively mismatching for HLA-DP results in much higher survival prognoses than for those that are non-permissively mismatched. So again, this is ways that we can find more donors for people, find mismatched donors for individuals if we have to, but they give less risk to the patient in their post-transplant. One of the other circles that I mentioned about in the Venn diagram at the beginning were the clinical factors to do with the transplant outcome and how we could use those to improve outcomes for our patients. One of the things that Anthony Nolan implemented going back some years was that we changed our donor recruitment ages. So we now recruit donors between the ages of 16 and 30. And the reason for that is that we, along with other studies, have been able to show that outcomes for patients with younger donors are actually better than those who are using older donors. So on this graph here, you can see that those individuals receiving a donor under the age of 30, so that's the blue line at the top here, they have significantly higher overall survival than those individuals where their donor was greater than 30. And so we want to try and find a donor under 30 for all of our patients to give them that benefit in transplant outcome. As I mentioned, this is data that's been studied quite extensively worldwide. This was a study from a much larger international study, but it really does show the same thing as the Anthony Nolan data. How the individuals here have been broken down is slightly different, but the dark solid line at the top here represents those individuals in the 18 to 32 category, age group, and they have significantly higher overall survival than those where the donor was between the ages of 33 and 50, and then those individuals who are over 50 as well. So really, we do want to be finding as many younger donors for our patients as we possibly can. The reason why older donors make or have a higher risk of a poor outcome is not that well understood, to be very honest with you. There are lots of hypotheses, but they haven't really been shown to be very conclusively shown. But one of the more recent studies that has come out has actually indicated that there are significant consequences for certain post-transplant outcomes. So on this graph here, those individuals receiving a donor between the ages of 18 and 32, they are represented by the pink line here, where that's sort of the baseline risk. And what we can find here is that those individuals that receive a donor either in the 33 to 50 year category or over 50 on the right-hand side of this graph here, they have significantly higher risks of mortality post-transplant. And we believe that that may have something to do with an increased risk of acute GVHD, which you can see just here and just here as well. I mentioned Cmv matching in one of my previous slides, and Cmv stands for the cytomegalovirus. It's a very common virus. Many of us will have had it. In some parts of the world, up to 80% of the population may have had CMV at some point. CMV is quite challenging. It is a virus that can lie dormant in our systems, and it can actually reactivate at points in our lives where maybe our immune systems give them the opportunity to re-emerge, to reinfect us, if you like. For most of us, if you are a healthy or relatively healthy individual, you could encounter CMV and you'd have symptoms similar to a mild cold, maybe a bit of a sore throat, and your immune system would knock that out and you'd clear that quite quickly. However, for patients, CMV becomes a real challenge. They are immunocompromised. They cannot deal with the reemergence of infections as quickly as healthy individuals, but also they just might not be able to actually have the immune system to be able to deal with that reinfection. And so CMV is very important in terms of transplantation. We certainly don't want that virus reactivating in patients that aren't able to challenge it. And so what we try to do is to match patients and donors as to whether they have ever experienced this virus before. And here you can see that those individuals who have been matched according to their CMV serostatus, the blue line just here, their survival again is greater than those who had a CMV mismatched transplant. Sorry. One of the things that we're very keen to do is how we start to use some of these factors together. And here you can see this is a study that was published a few years ago, and we're looking at those individuals that are HLA matched versus those that are mismatched but we're now including cytomegalovirus or CMV matching as well. So here, the individuals that are HLA matched and CMV matched have the best overall survival. And within HLA matched individuals, that negative effect of the CMV mismatch can be seen just here. However, when we look at HLA mismatched individuals, while there is definitely the survival is not predicted to be as high as the HLA matched individuals, we can actually offset some of the impact of an HLA mismatch by CMV matching. And this has really led to a step change in practice in the UK, where individuals who have no other choice than to pick an HLA mismatched donor really will not proceed unless they can find a CMV matched donor. So this is a way we're able to start to use the information together and provide choices for donors and for choices of donors for patients who are in need of a transplant. I mentioned also the non-HLA genetic factors. So we know that as well as we are matching patients and donors for their tissue types, their HLA genes, we know that this isn't answering all of the questions, and it's not resulting in survival for every patient or complication-free transplants for every patient. So we believe that there is a role for non-HLA genetic factors in predicting how patients perform after transplant, and ways that we might be able to include that into which donors we're finding for our patients. And here's just a couple of examples that we've published on over the last few years. The first example here on the left-hand side of this slide is an example of a gene called NOD2. And there are some polymorphisms, 3 known polymorphisms within the NOD2 gene that are known to correlate with complications with conditions such as Crohn's disease and other inflammatory bowel disorders. And so it was originally hypothesized that actually if they're a risk factor for these inflammatory bowel conditions in diseases like Crohn's disease, could they also be risk factors for individuals who've gone through transplants and may be incurring graft versus host disease of their gastrointestinal tract as well. And here what we were able to show was that that actually was the case. And we found that those individuals who were receiving transplants from individuals with a wild type, so the normal form of the gene, their survival was significantly higher than those who received a transplant from a mutated form, from a donor with the mutated form of the gene. So here's an example of how a non-HLA genetic factor could also impact on transplant outcome. And then in more recent years, this was a study published by one of our PhD students last year. Here we were able to see how the receptors on a family of cells known as the natural killer cells are actually able to predict transplant outcomes. So here, donors with certain characteristics of their KIR genes, the killer cell immunoglobulin-like receptor genes, on the surface of their NK cells, they were able to have a much better overall survival than those with different combinations as well, different patterns. So just to summarize a little bit really here, how the Patient Donor Project has actually changed practice both in Anthony Nolan and nationally and internationally. One of the really fantastic things about the Patient Donor Project is that we translate our findings into practice as soon as we can. So that's either changing how our search teams are searching for donors, who we're putting onto the register, or how we're typing patients and donors at the point of them either joining a register or needing to have a transplant. The 6th HLA gene that I've mentioned in a couple of our studies, DPB1 here, we have actually been routinely including that into our donor typing for nearly 10 years. And we've been using that actively for many years, which I think led to great benefits for our patients as well. Excuse me. We have responded to the data on donor age from not only our own studies, but international studies as well. And we've responded by reducing the minimum age to join our register. But not only that, we actively try and recruit younger donors. So we go into schools to talk about the importance of donation of all different kinds, not only of stem cells, and obviously working with fantastic groups like you, we're able to advertise and to inform and educate students at university as well as to the importance of being donors. Just some statistics actually. Because of this, the average age of joining our register is about 22 years, and the average age of an anti-neonatal donor is 27 years. So we really are finding donors for younger donors for our patients. On top of that, we're actually able to recommend that individuals should be, what level of matching individuals should be aiming for. So we have actually tried to implement this as well. We're looking for individuals that are 12 out of 12 HLA matched for all of our patients. But if not, which mismatches we should be avoiding, and made recommendations on avoiding HLA mismatched and CMV mismatched donors in combination. And I'd just like to finish just by acknowledging all of the transplant centres in the UK that actually work with us on this particular study. That's it from me. I'm more than happy to take some questions if there are any. I think I've bamboozled everybody with science this afternoon. It's a lot to take in. Just give it another minute or so for some questions if anybody has any. If not, I'm the kind of person who also, I often can't think of questions on the spot, but then they come to me in about 20 minutes after the presentation's finished. So if you do have any questions, please feel free to get in touch with our Marrow team, and they'll forward them on to me, and I'll get back to you with the answers. Okay. Oh, so we have one question. Is there a possibility for 10 out of 10 matches to be rejected by the patient? And how is this avoided if so? It's a really good question, and the answer is yes, there is a chance that a 10 out of 10 match may not be successful. What we try, or what transplant centres try and do, is to always aim for the 10 out of 10 match, because that gives us the best chance of there not being a complication. But patients are monitored very closely post-transplant for various different factors, including the engraftment of the cells themselves. So they have to, for many centres, the patient won't be allowed to go home before they've reached a certain number of cells within their peripheral system that indicates that the graft is actually taken, it's engrafted into the patient. And then patients are monitored very closely with their transplant team to keep an eye on complications post-transplant. Some of their treatment prior to the transplant will try and avoid complications. So many patients in the UK receive a treatment known as T-cell depletion. And what that does is it actually knocks out the T cells in the donor graft as well as in the patient, and that minimises the chance of there being a graft versus host disease response in the immediate post-transplant period. And they will also receive medication for a period of time post-transplant that again just suppresses the immune system to stop the post-transplant complications happening. But what we're hopeful for is that we do understand that these individuals who are receiving a 10 out of 10 match, they don't always have the best outcome, that they do incur complications. And the aim of this project really is to try and understand why what else should we be matching for? What else should we be doing for these individuals that we have given? I'm not that keen on the term perfect match. I'll do the little bunny ears. Because we don't actually know what that means, and people who do have the perfect match don't always have the perfect post-transplant outcome, and we need to solve that because it's not good enough. And so that's the aim really of this project, is to really understand more about that. And we can learn a lot from individuals who haven't had the perfect match, who haven't had a 10 out of 10 match transplant, but actually are doing very well post-transplant. They're not, haven't only survived, but they're thriving. And we want to understand, again, where genetics has played a role in that, and the transplant characteristics of that. How can we make that available to every patient? You're welcome. Any more questions? Okay. Shall we stop the meeting there then?